So today we'll finish our second lecture, and it will be the last lecture of the genetics. First, we will start with trinucleotide repeat disorders. In order to understand that, you have to know what is trinucleotide, right? So we know that in order to make our protein, we go through processes called transcription, where we make RNA, then translation, where we make protein. So protein chain contains of amino acids. Each amino acid have its own code, trinucleotide code on RNA and DNA, right? So this trinucleotide code can repeat from 3 to 55 times, and it is normal. But when it's more than 55, it is pathologic. When it's more than 200, it develops disorder, right? And we have 4 disorders that we have to know for USMLE. It is Huntington disease, myotonic dystrophy, Fragile X syndrome, and Friedreich ataxia. So when these repeats are actually increased, it happens in gametogenesis. We have like coding and non-coding regions that can be increased. Like for coding regions, it happens in spermatogenesis and it's associated with spinocerebellar ataxia, which is also trinucleotide repeat disorder, Huntington disease, right? Whereas non-coding regions increase in oogenesis happens in myotonic dystrophy, Fragile X syndrome, and Friedreich ataxia. So these disorders tend to show anticipation, which is where you increase disease severity and decrease age of onset in successive generations, right? We already discussed about that. So we actually know exact code for each disease. For Huntington disease, it is CAG. For myotonic dystrophy, it is CTG. For Fragile X syndrome, it is CGG, and Friedreich ataxia is GAA. And how do you memorize that? For example, these 3 have similarities. In their code, they have similar first and third letter. It is C and G, right? You memorize that. Then letter in the middle varies. For these letters in the middle, we have mnemonics. For example, Huntington syndrome, when you hunt, you catch something and put it into a cage, and therefore cage, right? A in between. Myotonic dystrophy, it contains T, and therefore CT in between G, right? Fragile X syndrome, it contains G, and therefore CGG, right? Then Friedreich ataxia, it is ataxic gait. It is exception from these 3. Ataxic gait, and you remember GAA, right? Gait. So then we can use these letters to memorize key features of each disease. For example, for Huntington disease, we have C for caudate has low, ACH, which is acetylcholine, and G GABA, right? Caudate has low acetylcholine and GABA, and therefore these patients have mood swings, tremor in their hands, and sometimes ataxic gait, etc., right? Myotonic dystrophy is CTG, which is cataract, right? Topy, which is early balding in males, gonadal atrophy in males, and reduced fertility in females, right? And again, myotonic dystrophy will be proximal muscle weakness when you cannot stand from your chair or go up on stairs. Fragile X syndrome, Why it called fragile X? Because, for example, you have chromosome, and this CGG genes located here, and when its amount increases, it becomes methylated, and it will be more condensed, right? And when it becomes more condensed, it seams this area, and therefore ends of this chromosome are prone to breakage. Therefore it's called fragile X syndrome. And fragile X syndrome is CGG. It's like C chin, protruding big chin, then giant ears and giant gonads, giant testicles in these patients, right? It is very recognizable in the stem for fragile X syndrome. Then Friedreich ataxia, again we know already, ataxic gait, right, for GAA. So let's move to the example here. An 11-year-old boy is brought to the pediatrician by his mother for a follow-up examination. Physical examination shows everted ears and a long jaw. Testicular volume is increased. Karyotype analysis shows an expansion of 650, right? More than 200. CGG, again CGG, it is fragile X syndrome. So repeats within the fifth untranslated region of the FMR1. FMR1 also associated with fragile X syndrome. Which of the following is the most likely effects of CGG expansion? Again, we already said that methylation will happen on this region, right? So let's look at the answer choices. First is decreased transcription. Transcription is when you make RNA from your DNA. Yes, this will happen when you will methylate the region because you will turn it off, right? Okay. Decreased translation. Technically it will happen, but first what will happen with decreasing transcription? Because we work with DNA, not RNA, right? Therefore, it's not our answer here. Hypermethylation causing increased gene activation. Hypermethylation, yes, it will happen, but we will decrease gene activation, not increase it. Impaired mRNA splicing, no, we're working with DNA. Again, impaired 5-methylguanosine caping of mRNA, no. It is like post-transcriptional modifications. We're not working with RNA here. Next, let's talk about autosomal trisomies. So when we get trisomy, when there is non-disjunction on the process of meiosis or mitosis, right? So here we have an image of meiosis 1 and meiosis 2. In meiosis 1, if there is non-disjunction occurs, we have like 2 gametes with trisomy and 2 with monosomy, whereas in meiosis 2 we get 2 normal, 1 monosomy and 1 trisomy. So for autosomal trisomy, we know 3 disorders. It is Down syndrome, chromosome 21, Edwards, chromosome 18, and Patau, chromosome 13. You see that Down syndrome is most often seen and is more benign than other 2. Why it happens? Because this chromosome is smallest chromosome in our body in general and in comparison to these 2 as well. Why it is smallest? Because we know that first one is the biggest and we go second smaller, third smaller, and first it was determined that we have like 22 autosomal chromosomes, 22 somatic chromosomes. First it was determined that 22nd was the smallest one, but then it was confirmed that 21 is actually smaller. but it was too late to change it, so we just know that 21st is the smallest one, and therefore it's most benign and least and more common than other two. So to memorize which chromosome associated with which exact disease, we have mnemonics. Like for Down syndrome, it starts with D, and therefore we use drinking age in US is 21. Edwards syndrome starts with E, election age is 18. Patau syndrome starts with P, puberty at age 13, right? And also for these diseases, we use screening in first trimester and second trimester. In first trimester, like when there's 10th or 13th weeks of gestation, we can use it. And second trimester is when 15 or 22 to 22 weeks of gestation, right? So how do we look at this? For example, most often seen 21st chromosome, which is associated with Down syndrome. We see markers beta HCG and Papa A. I call it Papa, right? These markers for second trimester, which is HCG, inhibin A, estriol, and alpha-fetoprotein. So how do you memorize this? You just memorize that for 21 chromosome, markers that contain H in it go high, go up, right? And you see that beta HCG, HCG inhibin contains H and therefore it's up, and others goes down. You just memorize like this for 21 chromosome. And for Edwards syndrome and Patau syndrome, it's always down or normal, right? It is easy to recognize them in both trimesters. So let's look at the examples here. 45-year-old female, 9 weeks gestation, Cardionius Willis sampling was performed, like it is will confirm actual diagnosis, right? Fetal trisomy 21. If a tree screen was performed prior to CBC, what were the most likely errors for serum levels of pregnancy associated plasma protein, PAPA, beta HCG, and fetal nuclear translucency? We said if there's letter H present, it will go up, right? And if there's no H, it will go down. Fetal nuclear translucency you see when you ultrasound this patient, right? Okay, let's go to the next question. 45-year-old female, 17 weeks gestation, concerned about risk for Down syndrome. If the fetus does in fact have Down syndrome, what are the most likely errors for the serum levels of alpha-fetoprotein, beta HCG, estriol, and inhibin A? Again, alpha-fetoprotein, no H, down. Beta HCG, there's H, up. Estriol, no H, down. Inhibin with H, up. That's it, right? So now let's talk about Robertsonian translocation. So what is translocation? When you take one arm of the chromosome and translocate it to the other part of the chromosome. Here you see like chromosome 14, for example, and 21. And this happens with acrocentric chromosomes, which means their centromere located too close to one end of the chromosome, and therefore they have short arm and long arm. And when translocation happens, you combine two short arms, which will disappear, and two long arms together, and you will get something like this eventually, right? So Robertsonian translocation is one in which effectively the whole of a chromosome is joined end to end with another, like this here, just their long arms, right? This type of translocation involves only chromosomes 13, 14, 15, 21, and 22 because the ends of their short arms have similar repetitive DNA sequences that predispose to their fusion, right? Because they have short arms and long arms, these short arms don't really have much information in it, and all this information is located on long arm, and therefore it will predispose to certain trisomy-like features, right? So for example, here we have chromosome 14 and chromosome 21, right? So this one is where Robertsonian translocation occurred. It contains two long arms of each chromosome. And I told you that one long arm can show all the features of normal chromosome 21. So we have, for example, normal 21 and 14 here as well. So we have possibilities for meiosis, which then can make certain types of digotes when it will combine with normal gametes, right? So for example, if we have this gamete where we have chromosome 14 and 21, for example, it will combine with another, this type of normal gamete, and make normal zygote. When we combine pathologic with translocation, technically we have normal 21 and normal 14 here, right? When we will combine it with normal zygote, we will have balanced translocation, so we will not see pathology in this child. Then when we combine, for example, here we have normal 14, again normal 14, technically normal 14, and technically normal 21. So we have too much 14 here, because normally we have only one 14 and only one 21. Here we have two 14 and one 21. Therefore, we have trisomy 14 here. Like when you look at it, you have 3 14, right? And 2 21s. Then when there's only one 21, you get monosomy 14, because you get 14 only from one parent, from normal parent, right? So when you get 1 14, again, you develop monosomy 21 because you get 21 only from one parent, normal parent. When you get translocation, translocated chromosome, let's say normal 21, 2 21s, technically normal, right? And 1 14, you get trisomy 21. Like, if you look at it, there's 2 21s technically and 2 14s, but this one will present fully like normal 21, therefore it is trisomy 21 here. So let's look at the example here. A 3-year-old girl is evaluated for conductive hearing loss, okay, due to Eustachian tube dysfunction and middle ear fluid collection. Physical examination shows slanted palpebral fissures, and single palmar crease. So one of the patient's eye is shown both under visible and near infrared wavelength. Fluorescent in situ hybridization shows cellular uniformity of karyotype, right? So the girl's mother is currently 17 years old. Which of the following is the most likely mechanism for the patient's condition? So we know that palmar crease and slanted palpebral fissures is associated with Down syndrome. Here on the images we can see brush filled spots here, you see, and here, which is also associated with Down syndrome, and Eustachian tube dysfunction and middle ear fluid collection called glue ear. It also associated with Down syndrome, and it will lead to conductive hearing loss in this patient. So let's look at the answer choices here. VT1 gene associated with Phillips tumor, right? Therefore, it is not our answer here. Mitotic non-disjunction is actually like 95% of cases of Down syndrome associated with it. And this happens when maternal age is more than 30 age, right? And our patient is 17. It is not the case here, therefore. Then we assume there is other cause for this patient having child with Down syndrome. It is either Robertsonian translocation or mosaicism, somatic mosaicism, right? So post-fertilization mitotic error is refers to somatic mosaicism. So we said in the stem that there is cellular uniformity when we made fluorescent in situ hybridization, which means all cells are similar, right? Therefore, answer D, which is mosaicism, is not the answer here. And translocation is right answer, Robertsonian translocation. Like this one here will happen in this patient, like trisomy, technically trisomy 21, where you have 1, 2, and 3, like only long arm of it, but technically it contains all of the feature of 21st chromosome, and therefore trisomy 21 technically because of Robertsonian translocation. And last one, there is deletion of certain parts of the chromosome. For example, when you delete short arm of chromosome 5, you develop Criduchat syndrome. And I have mnemonic here. You can pause and watch it back and memorize it. And then there is deletion of long arm of the chromosome 7 where you develop Williams syndrome. So for its features, you can look at this image here. So that's it for this lecture.